At the Gurdon Institute, we're interested in fundamental mechanisms involving non-coding RNA. Our genome is given at birth, but how the genome is utilized within our body is driven by a process called epigenetics. We discovered that RNA molecules in the model organism C. elegans can directly affect the epigenetic landscape. This is exciting because regulating the epigenetic landscape can have effects that last beyond a generation and may be important in understanding heritable human disease. What has really revolutionized the analysis of non-coding RNA is our ability to sequence DNA. Nowadays, we can use a machine as simple as this to get really deep insights into this exciting biology. One particular non-coding RNA that we've been studying for many years, the LED7 microRNA, is a direct causing agent in cancer. This raises the exciting possibility to develop new anti-cancer therapeutics that are themselves based on RNA. With access to many more genomes now, we can study the role of non-coding RNA in evolution. To do this, we have chosen an amazing system we refer to as Darwin's pond. Large lakes in Southeast Africa, where many close related species of fish live, the so-called cichlid fish of the Rift Valley. If you look at their phenotypes, they are very, very different, but the level of the genome, they all look very similar. By studying these fish, we hope to understand something about maybe how the human genome evolved. I'm very grateful for having been awarded the Robert Hooke Medal by the British Society for Cell Biology, because it brings me back to my early childhood when I discovered with my first microscope how amazing biology can be. What is really so exciting in science is the ability to interact with very creative people of different disciplines. It's important to support fundamental research because we never know how it might transform our lives tomorrow.